Today, we will be discussing the different methods of cell communication. First off, we will discuss the G-protein receptor system. As you see here, we have the plasma membrane with the G-protein linked receptor embedded in the membrane. Next, we see the inactive G-protein. This G-protein is inactive due to the presence of glucose triphosphate, or GDP, as opposed to glucose triphosphate, or GTP. Here, we see the approaching signal molecule. This molecule is a protein and is referred to as a ligand. It will attach to the receptor. The signal and receptor are specific to each other. Next, the inactive G-protein will travel to the receptor and will become an active G-protein with the GTP instead of GDP. The receptor has given this protein an additional phosphate that causes its activeness. Next, the active G-protein travels away from the receptor and meets another inactive G-protein. The original active G-protein will transfer a phosphate to make this inactive G-protein active. As this happens, the original G-protein will become inactive. This process occurs a few more times. This is referred to as the signal transduction pathway. At the end of the pathway, an active G-protein is approaching an inactive enzyme. The G-protein will attach to the inactive enzyme and activate it. The enzyme now has ATP. The enzyme will convert this ATP into cyclin AMP, or CAMP. This CAMP is converted into a protein kinase. This protein kinase will travel to the endoplasmic reticulum. The kinase will trigger a response in the ER, and the ER will release calcium ions into the cell. After this response is complete, the G-protein detaches from the enzyme. The enzyme will become inactive once again. As this process occurs, an organic phosphate will be released from the G-protein, and the G-protein is inactive once more. Here we see the receptor again. Since the cellular response has been completed, the ligand can now detach from the receptor. Now let's move on to the second method of cell communication. This method involves tyrosine kinase receptors and therefore is called the tyrosine kinase receptor system. Here we see two inactive tyrosine kinase receptors. There are 3 tyrosine groups attached to each of them. A signal protein attaches to both of them simultaneously. This triggers the receptors to move and attach to each other. This double structure is known as a dimer. Here we see 6 adenosine triphosphate, or ATP groups, approaching the dimer. Each of them attach a phosphate group to a tyrosine on the receptor. These ATP groups are now ADP groups since they have lost a phosphate. The receptors are now active due to the presence of phosphates. Now we see relay proteins approaching the receptors. These relay proteins are currently inactive. Now they take a phosphate from the tyrosine kinase receptors. These proteins are now active. This active relay protein will transfer the phosphate to an inactive protein kinase to activate it. The ADP attached to the kinase is now ATP, making the protein kinase active. Then this active protein kinase will transfer a phosphate to another inactive protein kinase, making itself inactive and the new kinase active. This process repeats once more. This is known as the signal transduction pathway. Here we see an inactive protein. The activated protein kinase will transfer a phosphate group to the inactive protein to activate it. This active protein travels to the endoplasmic reticulum and triggers the release of calcium ions into the cell. This is the cellular response. After this occurs, the signaled proteins are released from the tyrosine kinase receptors and they separate once more.
Cholera is one of the diseases caused by errors in cell communication. Here we can see the active G-protein approaching the adenylyl cyclase. The G-protein then transfers the ATP to the enzyme, and the enzyme will convert it to cAMP, or cyclic AMP. This will then activate a protein kinase which starts the phosphorylation cascade. The protein kinase will open the gated ion channels as a cellular response, allowing molecules of sodium and chlorine to exit the cell. Water molecules remain in the cell, and back at the receptor, the ligand unbinds from the receptor and the receptor is inactive. Now we view an epithelial cell affected by cholera. The cholera toxin signal protein binds to the ganglioside protein receptor. This new signal protein comes from the cholera bacteria Vibrio cholerae. The toxin then enters the cell and is converted into a G-protein. The G-protein binds to the enzyme adenylate cyclase. The enzyme then activates and converts ATP to cyclic AMP. The CAMP then activates a protein kinase. This activated protein kinase will open the gated ion channels and release molecules of chlorine and sodium. In addition to these, water molecules also exit the cell. This is due to the signal and receptor being bound together, meaning the signal never leaves the receptor, and therefore the water leaves the cell indefinitely.